The first thing I want to start by saying is that nanomedicine has transformed drug delivery. And it's done this by basically allowing us to package therapeutics into lipid nanoparticles. And we can take these lipid nanoparticles and functionalize them with antibodies specific to our target site. So say we want to deliver some kind of therapeutic to T cells, we can add antibodies that bind the T cells onto our lipid nanoparticle. So when we inject that lipid nanoparticle, it's basically heat seeking for T cells. It seeks those T cells out, it delivers a therapeutic. And this is compared to passive distribution. So for example, if we're administering a drug via IV, and we just let it passively flow throughout the body, you can get a lot more of your drug delivered using the targeted therapy. So this opens up all sorts of exciting opportunities for new treatments and new therapies. But because of our healthcare system, this access to these new therapies isn't exactly equitable. And that's because medicine and healthcare are centralized. So what I mean by this is that there are factories in centralized, well-resourced areas where drugs, vaccines, proteins, diagnostics are produced. Then they're shipped around the world at 4 degrees or colder, as we're seeing with a lot of the COVID vaccines right now. They're sent to hospitals and clinics where patients then go to receive care. This model works well in urban, well-resourced places like Toronto, where I am, because I can look out my window and I could see a hospital right outside. But if you're someone living in a remote community or a less resourced community, there are a lot of challenges associated with this healthcare model. So one of the focuses in our lab is kind of using synthetic biology to decentralize healthcare. So for this project, we kind of asked, okay, can we decentralize nanomedicine? So in other words, can we build a portable device that can take nanomedicine production out of the factory and into the field? So in the previous slide, I showed a factory where drugs are being made, then shipped across the world to where they're needed. Can we instead have a benchtop device that we can just press a button and we get a functionalized lipid nanoparticle with whatever antibody we want to send it to whatever target tissue in the body we want that carries the drug of interest. Basically just on the benchtop, you hit a button and you get that coming out. So this is building off of work that we've done previously where we built a portable device for protein expression. So what we used here was something called freeze-dried cell-free protein synthesis. And I'm not going to dig too far into this, but what this is basically a reaction pellet that contains all the enzymes and all the ingredients necessary to produce proteins. But what's cool is we can freeze dry this, we can store it at room temperature. So when you rehydrate it with water, the protein starts to be expressed. So what this device looks like is you have a cassette containing the freeze-dried cell-free protein synthesis reaction pellet and plus the DNA template of whatever you want to produce. You plug that into the device, it gets rehydrated, protein is expressed, purified on the device, and then after a few hours, you have a purified protein. So with this, we can make things like GFP, therapeutic antibodies, enzymes, antibiotics, and more. So with this project, we're thinking, can we take our device, can we expand its use to produce functionalized LNPs? So first of all, can we express and purify an antibody just using the normal system that we've already put together? Can we have the DNA that makes this antibody, expresses it? Can we enzymatically conjugate that antibody to our lipid nanoparticle? And then can we encapsulate our drug inside that lipid nanoparticle so that now our device actually creates a nanomedicine for you? So again, what this looks like is you have your DNA, your freeze-dried cell-free reaction, your LNPs, your lipid nanoparticles. You have this in a cassette that goes into the device, and on the other side, you get this functionalized lipid nanoparticle with all your antibodies and your drug loaded. So as a proof of concept, we wanted to use CAR T-cell therapy. So this is a really good figure that kind of just shows how CAR T-cell therapy is traditionally done. So basically, CAR T-cell therapy is a cancer therapy where you take the patient's T-cells, which are the cytotoxic immune cells, and you engineer them to basically superpower it against the patient's cancer. So step one is you remove the blood from the patient to get the T-cells. Then you engineer those T-cells outside of their body. So you have a petri dish, you're adding in your gene, so that your T cells express a CAR, a chimeric antigen receptor. And what a chimeric antigen receptor is you have a single chain variable fragment or an antibody on the outside targeting a patient's cancer that's connected to the normal signaling domains of the T cell. So when it binds onto your cancer cells, the signaling domains are activated and the cell becomes cytotoxic. Then you grow millions of these cells up and you introduce them back into the patient. So for this project, we were wondering, can we make LNPs in our device that target T cells in the patient's bloodstream? So basically, this LNP will carry the CAR cassette, the actual gene to code the CAR into the T cell, gets injected into the patient's blood. The antibodies guide that LNP to the T cells in vivo, so in their body, deliver the CAR, basically making CAR T cells in the body instead of outside. So instead of having to take those cells out of the body, engineer them outside, instead we're putting a lipid nanoparticle into the patient that targets their T cells to deliver the CAR to the T cell. If that makes sense. So the project outline: step 1, develop molecular tools needed for production of functionalized LNPs. So we're conjugate anti-T cell antibodies, or CAR cassette. Step 2, integrate these molecular tools into the device. And then step 3, validate the ability of our nanoparticles to generate CAR T cells using in vitro and in vivo studies. So our first step was to develop some T cell specific antibodies. So remember, we want these T cell specific antibodies to be attached to the outside of our lipid nanoparticle, so our lipid nanoparticle can seek out T cells once it's inside the patient. So I got a couple sequences for CD3 and CD8 single chain variable fragments and nanobodies, which are basically just different types of antibodies. And CD3 and CD8 are T cell surface antigens. So these antibodies will bind onto a T cell. I express these within our protein synthesis device using cell-free protein synthesis. And this is a Western blot just showing that they did express. So it kind of just proves that our technology works and can support synthesis of these antibodies. So our best performer was an anti-CD3 nanobody, so we went forward with that. So now we have our antibodies being expressed, and now we need to attach it to our lipid nanoparticle. So for that, we need something to conjugate those two things together. So I chose sortase for this. Sortase is an enzyme. It's a transpeptidase. What it does is it covalently bonds two proteins together. So one of those proteins will have a C-terminal LPETG amino acid motif, and the other protein will have an N-terminal polyglycine. So there's a bunch of glycines on the end. So here's our lipid nanoparticle with our polyglycines, and here's our antibody with the LPETG domain. I add sortase, transpeptidation occurs, and we have now a covalent bond between the two. This figure, I just want to show you that we can actually show this conjugation works. So this band here is our CD3 nanobody. In this lane, I've added another protein with a polyglycine, and I've also added sortase. So what we should be seeing is two proteins being connected together and a size shift indicating that there's a conjugation between those two. And that's what we see, an increase in size showing that our two proteins have been put together. So this shows that sortase is working. So we have the antibodies expressing, we have our conjugation scheme working. And so for the next steps, we need to integrate these into our hardware to actually make the device work. We need to test our LNPs made on the device with T cells in vitro and then in vivo with mice. And of course, I want to speak to some of the limitations of this project, mainly that this is really proof of concept, right? So it's difficult to implement QC, quality control, in something like a portable on-demand device, right? And quality control is really important for making therapeutics. So, we understand that this is very proof of concept and that there are necessary steps that we're going to have to think about in the future and as to how we're going to control quality control. So before I deliver kind of the main key messages of this work, I did want to thank my team and everyone involved in the project. Dr. Masoud Narouzi, who's worked on this project alongside with me, and Dr. Ryan Phobel and Fahim Massoum, who have both done a lot of work on the actual device itself. My PI, Professor Keith Pardee, my committee members, and of course, funding sources and assistance from a variety of different sources. So the key message here is that we're working on making a portable, on-demand, abiotic platform for benchtop synthesis of functionalized lipid nanoparticles. So again, we have DNA that encodes the proteins we want to make, so our sortase and our antibodies. We have a freeze-dried cell-free reaction, our LNPs, goes into a cassette, plugged into the device where it's rehydrated, proteins are expressed and purified, and then our LNP is assembled with those antibodies that will guide it to the correct site, loaded with the CAR cassette and everything. So by doing this, we're able to improve accessibility of nanomedicines to remote and low resource communities, as well as for researchers. So you can have it sitting on your benchtop if you want LNPs that go to a specific tissue site. You just need a different DNA for a different antibody, and you can just play around with that as much as you want. So thank you for listening, and I look forward to answering any questions.
Excellent. Thank you very much, Quinn. So I'm pleased to open the floor to questions from the audience. Please either submit a written question or raise your hands. And while we give people a moment to do that, I'd like to ask a question, Quinn.
Sure.
I'm curious about, so once this is out in the rural communities or in researchers' labs, what are you expecting, how long are you expecting this process to take?
Yeah, that's a good question. So it depends. There are, of course, multiple steps here. There's a step where you're actually expressing the protein. There's a conjugation step and an encapsulation step. So for protein synthesis, for really well-folding, easy to behave protein, we get typically peak out around 3 or 4 hours. So someone like GFP, after 4 hours, it's kind of saturated. With some more difficult proteins, we see 6 to 8 hours. You get a relatively appreciable amount within the first 2 to 3 hours. So we can have a pretty quick synthesis of the protein. The conjugation typically takes anywhere from half an hour to 1 hour. So we'd have maybe 3 hours for protein synthesis, an hour for conjugation, and then after maybe 5 hours allowing for all the user input, you probably have your ready to go lipid nanoparticles after that amount of time.
Great, thank you. Hayden, are there any other questions?
Yes, we do have one question in the chat from Grace. Thank you for the great presentation. I was wondering, how long does it take to form the nanoparticles, and how stable is the end product?
That's a good question. So we haven't quite gone to the actual nanoparticles testing stage yet, so I'm not a professional yet in this area. But the actual encapsulation process is pretty quick. Basically, you have your drug is sitting in an aqueous environment, and then your lipids are in a non-aqueous environment. When you mix them together, they kind of quickly encapsulate. So it's a relatively quick process. In terms of stability, I think that depends on what you have conjugated to the nanoparticle itself, as well as what you have loaded in the drug. But for this therapy, it would be something that you're not necessarily needing to store these, right? So you need lipid nanoparticles, you would be producing them and then inject them as soon as possible. So stability would probably be long enough for this to actually be useful. That's a good question though, thank you.
Excellent, thank you, Grace, for your question. Hayden, could you read the next one?
Yeah, so the next question is from Miffy. Could you talk a little more about the sortase conjugation? What are the benefits of this over the traditional thiol bioconjugation?
Yeah, so the enzyme-based conjugation is quite simple. First of all, what's great about it is that it's really site-specific, right? So you have your LPTG and your polyglycine, and you know it's going to attach at that point. There are other methods for conjugation that are chemical-based, like the one you mentioned, like a malamide-thiol reaction. Those necessarily, they typically require mixing of reagents and maybe heating, cooling, might take a little bit longer. So for a device, we want minimal user input that can already produce proteins. A really simple method is for us to use a sortase-based reaction, right? Because sortase is an easy to produce protein. We basically express it alongside our antibody. And then because we have those domains already on our proteins, it's so easy for them just to become conjugated. So we just found it to be the most simple, easiest for the user, makes sense with the device. So we chose to go with that. That's a good question. Thanks.